So I want to continue a little bit on the topic of microbial diversity. And this is a good intro into microbial structures, which we're going to spend most of the day talking about. Because some of the structures are vastly different between the big groups of microbes. So just to review from last time, the current organization of how microbial groups and actually all life, the way that organization is done now is by sequencing ribosomal RNA genes and comparing those genes that all life has, and the more similar those genes are, the more similar the ancestry of the organism. And that puts us into, well, us literally, that puts us into the group with eukaryotes, which contains most of the life forms you've talked about before now. So we have domain eukarya, domain archaea, domain bacteria. And in this class, we're actually going to be focusing almost all of our time on domain bacteria. I'll sometimes talk about archaea, but mostly we'll be using domain bacteria. And in the lab, we're going to be largely working with gram-positive bacteria and what we call proteobacteria. So the gram-negatives are in class proteobacteria. And one other one that doesn't fit well into these two groups. But I want you to notice in domain archaea what kind of names they have. So Pyrodictyum, Thermoproteus, Thermococcus, Pyrolobus, Methanopyrus. What does that bring to mind?
External heat, pyrophore, methane.
Heat, fire, methane. Yeah. If you haven't heard much about archaea, it's because they have been discovered kind of relatively recently, because they tend to live in really extreme environments that are hot. They might make methane. Some of them are using sulfur as their food source. And the way that some of these microbes do their day-to-day life is really, really weird compared to how you might have thought about how life is powered before. So I want to spend a little bit of time on this picture right here, because this is something that you just don't see until you study microbiology. And what this is doing is dividing microbes into two big groups based on what's their energy source. And so in the past, you've studied light as an energy source in that plants are doing photosynthesis and using light as their energy source. There are also some eukaryotic algae that do this, and there are a lot of bacteria and archaea that do this too. They use light as the energy source. And because they use light as an energy source, we call them phototrophs. So photo for light, phototrophs, and they use light to make ATP. The other metabolic lifestyle that you're probably familiar with is what this book calls chemoorganotroph. So they're using chemicals for the energy source, and not just any chemicals, but organic chemicals. And this is what we think of as food. So we are chemoorganotrophs. We use organic chemicals for our food, and our body prefers glucose. What do plants use for food?
Also glucose.
Yes, they make glucose and they use it for food, but they make it themselves. So think back when you're very, very little and you learn what plants need to grow. What do they have to have? Sun, water, mineral nutrition, which as a child you learn as soil. And there's something else they use for their carbon source that you don't learn about until you're bigger. What's their carbon source?
CO2.
CO2. So if CO2 is a carbon source, it's not really considered food, it's a building block. If you're using chemicals for energy, that's a different situation and you're an organotroph. This is the group I want to talk about that so far only microbes, certain microbes do this. They're using chemicals for energy, but they're using inorganic chemicals. Not sugars, not bigger, complex molecules, but chemically tiny stuff like this. Hydrogen gas, that is crazy. Hydrogen gas is the chemical energy source for a lot of microbes in the world. So is hydrogen sulfide. Different forms of iron, ammonium. That's very weird, but not for some microbes. That's their normal lifestyle. So they're using inorganic chemicals for energy. And they are usually using CO2 for their carbon building block. Chemoorganotrophs use organic chemicals for energy and for carbon building block. And all of the organisms that we are really used to studying that aren't plants have that lifestyle, like us. So what this means is that there are a large number of microbes as a group called chemolithotrophs, chemical rock eaters. Rock chemical eaters would make more sense, but eating this stuff for energy and using CO2 as their carbon source. I have up here the question, how does this relate to heterotroph and autotroph? Are those the terms you use like in previous classes? Our book doesn't use those terms. So heterotroph, autotroph, what do those generally mean to you? If you had to say what's a heterotroph?
They get their food from like other sources and not they don't make it themselves.
Yeah. They don't make their food. Autotrophs make their food. Heterotrophs eat their food. Part of the reason our book doesn't use those terms for microbes is it doesn't quite fit. So heterotroph, autotroph works well if you think about the world as plants, algae, and the regular food eaters. But in this class, we'll talk about chemotroph versus phototroph. And within chemotrophs, are they chemoorganotrophs or chemolithotrophs? Yes.
So would plants be chemoorganotrophs because their ATP comes from glucose and not directly from lime?
Would they be chemoorganotrophs? No,
It would be phototrophs
they are, because they're making the glucose
themselves.
using CO2 as a starting building block. So all of these are carbon-based life forms.
Mhm.
They're all built out of carbon building blocks, but where they get the carbon is actually I should add that on here too. So usually CO2 is the building block to get carbon for an autotroph, and an organic carbon source is the building block for a heterotroph. I hate when books use different terminology. But our book, since we're going to use it, chemolithotroph, that's really what I want you to really get out of this, is what does that actually mean? They don't eat food like we think of food as large organic molecules that have carbohydrates or something. Very different chemical metabolism. this next slide is actually the last one out of this slide set that I'm going to talk about today. And I don't often ever reference the Bible in this class because these organisms are not listed in the Bible, except maybe the one, there is leprosy and there is disease, but the organisms that cause them were not known then. But if we think of creation and the creatures on earth in a sense of wonder, these creatures are wonderful in their way, and the places that they can live are amazing. So this also is a vocabulary list for you. So I want you to know these terms, which are pretty easy. The only one that is not immediately obvious what it means is psychrophile, which is a cold lover. So all of these have phile from philos, lover of. Hyperthermo, those hyperthermophile live in extremely hot temperatures. As in, this is crazy, they cannot live below 90 degrees Celsius. That's remarkable. It's not that they just tolerate hot temperatures, they have to have hot temperatures, and we'll talk about why in a few minutes. And this is for this specific hyperthermophile. This specific psychrophile, Psychromonas, it got a good relevant name. If it's over 10 degrees Celsius, it will die. So the temperature in this room would kill it. It needs it to be cold. Likewise, there are acidophiles that live in a pH optimum of 0.7. I'm geeking out. That's remarkable. Alkalophiles, pH optimum of 10. This is closer to, the extreme of pH than this one is. We'll talk about that too later in the semester, why that is. A barophile, think of barometric pressure. They live best at 700 atmospheres. It also raises the question, how does anybody study these? Can you recreate these conditions in the lab? That's why they're hard to study. And then halophile, like halogens, they do not just prefer but need to be in 25% solute condition. So, number one, marvel at what life can do on the extremes. And the extreme conditions, when life can tolerate extreme conditions, it's in microbial form. And also recognize what these terms mean, which is not really difficult. Yeah?
Do we need to know like the specific species?
No.
Just will tell.
No. It would be fun to have, so the question was, do we need to know the specific species? No. Whenever you need to know stuff about specific species, it's usually in lab, and I'll let you know which. But in the future, if you see something like Natronobacterium gregorii, it's helpful to be able to pick out what is natro. That's the same root word as sodium. It lives in a really salty place. Something like this, it's named after the person that discovered it. Halobacterium salinarum. Even if you've never seen something like that before, you can sort of guess what it is. Halogen, saline, it lives in a really salty place. These, maybe I'll just quickly go through these two slides but not write anything on them. So I do need to write a little bit on these. So domain bacteria, these are the big groups. And within this group, I'll make just a few notes. This group is photosynthetic. In the anoxygenic style. We talked about that, what that means, right? Generally. They do photosynthesis, but they don't make oxygen. Cyanobacteria, you may have heard of those before. Those are photosynthetic in the oxygenic style. So they act a lot like plants do in their photosynthesis. Chlamydia, you may have heard of. It's in some special group. Not every STD microbe is in its own special group, but it has unusual structure and physiology. Gram-positives, we'll learn a lot about these in lab, but Staphylococcus, Streptococcus, most Bacillus species are gram-positives. And then the proteobacteria, some of the ones of those that you have heard of before are Salmonella and E. coli. And we're going to study those in the lab as well. That's just to give you an overview, and also to note that what we study in the lab is a very small, in lab here at King, is a very small representation of all of the types of bacteria that do exist. Green sulfur are a lot like green non-sulfur. They also do anoxygenic photosynthesis. Syphilis is a spirochete. And I think this slide I'm not going to talk about. I wish I had more time to talk about archaea. They're remarkable.